Everyone bear with me. I have the daunting task of representing a full.
Sorry,
Oh,
didn't show you, but this is the point
Yeah.
forward.
okay. Thank you. So bear with me, everyone. I have the daunting task to represent a full lab's research in 5 minutes. What my main topic and I want to talk about is our recombinant viral vaccine vector system. And I want to start off first by thanking all my lab members, what we call the virology team. They can't be here today. They're actually working in the isolation unit on my project, so I really need to thank them first. And of course, all of our funding agencies, NSERCO, MAFRA, CPRC, and PIC. So our lab is involved in quite broadly avian virology. We focus on plenty of topics. Just listed a few here. Pathogenesis. Our virus of interest is Fowl adenovirus, FADV. So we look at genomic studies, the immunology behind the virus, the molecular biology behind the virus, on top of course, my interest, Fowl adenovirus-based recombinant vaccines. So to give you a brief introduction, Fowl adenovirus belongs to the family Adenoviruses. These are large, double-stranded DNA viruses with linear genomes, and in the context of poultry, they have a worldwide distribution. They're quite ubiquitous, and exist in pathogenic and non-pathogenic serotypes. Some of you may know the disease these viruses cause, such as IBH, inclusion body hepatitis, and hepatitis hydropericardium syndrome, the latter of which is not seen in Canada. So I guess the most important part of my project is a recombinant virus vector system that was developed in the NAJ lab a couple years ago by previous students, which we have coined the FADMID system. So the FADMID system itself uses a non-pathogenic strain of FADV9. And what happened was a student was able to discover a region in the genome, at the left end, if you can kind of see that on the white board, that is dispensable for virus replication, which we can use to add transgenes, of foreign viruses or foreign bacteria that we could vaccinate against. So this is a dispensable region. And this region has enabled us to make plenty of recombinant virus vaccines so far for proteins maybe of not of interest, but with EGFP, or with proteins and genes of chronic diseases such as avian influenza, highly pathogenic HA, or Newcastle disease virus, hemagglutinin neuraminidase protein. And as you can see, we, again, with this laser pointer, you can see that we are able to produce these proteins with the virus, and we're able to infect a wide variety of cells. But specifically in chickens, we're able to mount an immune response, which is what I'm interested in. So my research question is, how can we optimize this overall expression? Because really a vaccine needs to be able to efficiently produce an antibody response to the disease. So my research idea came from what we do in human viruses. We're systematically comparing promoter and enhancer element expression to figure out how we can develop the optimized vaccine for not only the model system we're using, but for the tissue we're trying to vaccinate with, or tissue we're trying to mount immune response in. So I'm looking at a variety of promoters, both avian and human origin, listed here. Some of you are probably familiar with, for example, the CMV promoter, and enhancer elements that recently we've seen in the literature that can increase antibody expression 2 to 5-fold in a variety of animal systems, such as the WPRE element. And just to give you just some quick data, what we're noticing right now is that these promoters, over a course of 72 hours, for example, all demonstrate different levels of expression and different expression patterns. And this is interesting to us because it's interesting to us because depending on how we want to vaccinate, whether we want an early expression at 24 hours, or if we want a slow progressive expression, for example, with the EF1 alpha promoter, this can determine how we make our virus vaccines. So then our research in industry. Currently, we are studying the promoters and their activity both in vivo and in vitro, and our future research will continue to look at this virus at the molecular level, figuring out what the deleted region of this virus has done to the overall immunology behind it, and how we can develop these recombinant viruses into better vaccines. But really, why should you all care? When it comes down to it, the avian immune system responds very well to vaccination. And we know we have 100 plus certified vaccines on the market, and disease itself decreases performance and production. So if we can create better vaccine vector systems to protect against diseases that can be cheaper and more efficient, this helps everyone. So on that note, I'm done, and I will entertain any questions.
Okay, thank you, James.